All right, so this week we are going to be finishing up biotechnology and moving on to evolution. Um, and I just kind of wanted to use this lecture to set us up for the next section of biotechnology by reminding us of some of the previous ideas and also telling you something about me. So, we're going to talk about each of these little grainy images in just a second. But like I said, we are moving from biotechnology into evolution. And the reason I'm showing you these particular images, I'm not going to ask you to understand any of these necessarily, but this is work from my research. And for me, when I was in school, I thought it was very important in many instances to get an understanding of what the teacher likes, what the teacher is into. And this is what I like. This is what I'm into. I like data questions, but I really like where the idea that you can sequence a genome meets up with the idea of the evolutionary process and how that applies to ecology. And so, like I said, this is images from my research. I studied butterflies in addition to a few other things that we're going to probably be talking about during this lecture or the next lectures. But I just wanted to let you guys know, it can be challenging, especially within this context, but if I'm explaining something too fast, you can just email me and tell me, hey, Aaron, you were going way too fast. Because I do get excited about these things, and it is easy for me to gloss over things because I really like this stuff. So yeah, if you're struggling through the lectures, you can let me know. I can re-record things. I can answer things through email. But let's talk about what we're going to be doing today. Or rather, let's talk about things that you need to be remembering from the last section. So hopefully you remember what a plasmid is. Plasmids are just like these extra little segments of DNA that we can take and put into bacteria. And because we have a fairly robust understanding of the biology of these things, we know how the process of DNA replication works, we can actually take our own little segments of DNA, cut them up, and put them into these plasmids, and then put them into bacteria, and those bacteria will grow whatever protein we want them to grow. So what I'm saying here is these plasmids are extra segments of DNA that bacteria often use in nature. So bacteria will just encounter plasmids in their environment sometimes. And what we have done as biologists, as scientists, is we've taken and we've stolen this technology from them, and we're using it to our own ends. So plasmids can be used. You can take out a plasmid, modify it, put it back into a bacteria, and make that bacteria make whatever protein you want. So one of the tools that we use to accomplish this is restriction enzymes, which hopefully you remember are those little molecular scissors. So restriction enzymes cut at specific points. They cut at specific points in a plasmid or in any segment of DNA. And we use this fact, and we use the fact that they generate these things called sticky ends, which I'm not as worried about. But we use the fact that we can cut DNA in a very specific way, and we'll cut open that plasmid. We can insert our own gene where this thing has cut. So another technique that is very important for modern biology is the idea of polymerase chain reaction, or PCR. And what you need to know about PCR is that it is basically just a molecular copy machine. You can get a very small amount of DNA, like let's say you're a researcher and you find just a little bit of blood at a crime scene or wherever you are, and you can copy that DNA using PCR. PCR is a really big idea. It's been around since the 1980s. In fact, it utilizes the enzymes of archaea. It utilizes enzymes of organisms that are in really, really hot environments and allows us to manipulate DNA using those. But what you need to know is that PCR is this magnificent molecular copy machine. The next thing to sort of keep in mind is the structure of DNA and electrophoresis. So we talked about electrophoresis back before the exam, but what electrophoresis allows us to do is separate different segments of DNA based on their size. The reason that we're able to do that is because DNA has all of these phosphate groups, which wind up making the overall molecule of DNA be negatively charged. So we're able to use that negative charge to use electricity to move segments of DNA. Okay, so what do you need to know? Plasmids, extra pieces of DNA that can be inserted into a bacterium and allow us to make whatever protein we want from those bacteria. Restriction enzymes, these are molecular scissors that cut at specific points that allow us to cut open a segment of DNA and then paste in whatever we want to it. Well, not whatever we want, whatever segment of DNA we want. And again, you can have bacteria growing human genes. You can have bacteria making human insulin or human growth hormone. We also have this notion of PCR, which is this molecular copy machine. You can make copies of segments of DNA. I want you to know that DNA has a slightly negative charge, which allows us to put it through the process of electrophoresis. Electrophoresis allows us to separate different sizes of DNA. And we also have those enzymes that we learned about in the last section. And you might have been wondering, like, who cares? Why are we learning about this? This seems so oddly specific. The reason that we're learning about those, or that we learned about those, is not only does it have implications for cancer, but it also allows us to utilize many of these processes. And basically, knowing about those things is allowing us to push biotechnology forward. It's allowing us to basically, we don't invent biotechnology, we hijack it from the cells where it already existed, and we utilize it for our own ends. And so studying those enzymes and understanding how they work is it's pivotal for all of these processes and making them work. So these are things I want you to be thinking about as we finish up with the last section here, and as we move on to the next section, which is evolution. We're probably going to be posting lectures throughout the week this week, so watch out for that. Hopefully Canvas will notify you, and it'll just pop up and say, okay, new lecture is posted. If not, I'm also going to be posting announcements whenever I put out a new lecture this week.